Hello, everyone, and welcome with another session this evening of Virtual Global Spine. Tonight, it's my pleasure, distinct honor to host my partner and friend, Dr. Christopher Ahuja, who is not only a spectacular complex spine and deformity surgeon, but also one of the few in the world who is a true scientist, who is extremely prolific in the arena of stem cell research and neuroregenerative science. Tonight, he'll be speaking about and update us on the therapies and stem cell therapies for spinal cord injury. And this is one of the best talks I've heard in my career in that domain. And he's going to take us through a wonderful adventure. Dr. Ahuja, the floor is yours. And here's behind me is Dr. Khan, who's also going to tune into this spectacular session.
Thank you, Dr. Dodley. Appreciate it. Appreciate to yourself and the Virtual Global Spine organizers for inviting me. It's very kind of you. I'm just going to share my screen here and we'll get started. Perfect. Are you able to see that screen all right?
Yes, sir. It's spectacular.
Excellent. All right. So this talk is really formulated around trying to develop the next generation of regenerative medicine and neural stem cell therapies to regenerate the injured spinal cord. And this was work that I did in combination with Dr. Michael Failings, partly in his lab and partly as collaboration, as well as with many of the scientists that you see listed below there. One I'd like to mention is Dr. Mohammed Kazai, who's really an outstanding and really exemplary scientist who contributed significantly as well to this work. So I'll start off by just talking a little bit about spinal cord injuries and why they matter so much to all of us and to me in particular when I started seeing these folks in clinics. So what I realized very quickly is that spinal cord injuries, unfortunately, are quite prevalent, especially in our domain. They affect about 350,000 individuals across the U.S. I'm from Canada originally, so about 40,000 across Canada, over 1 million people across G7 countries, or about 4 million people worldwide at any one time. And folks are living, fortunately, longer and longer after these injuries. We're getting better at the medical aspects of care, preventing pneumonias and blood clots and treating some of the concomitant complications that occur. But that also means that people are living with these devastating sequelae for longer, and families and caregivers are the ones that are often taking on that role and responsibility of the care. There are currently no effective regenerative therapies for spinal cord injury. So when a patient comes to us in clinic and they say, I've done my rehabilitation diligently, what's next? How do I continue to get better? The answer that unfortunately myself and I think most of us have is nothing. There's nothing else that we can offer at this point that can really continue to improve your function. Whatever you have now is likely what you're going to have for the rest of your life. And spinal cord injury, it's an interesting field, especially in Europe and the US, because it has orphan disease status. So that means that even though it has a relatively high prevalence, because it's such a devastating condition with no treatment options, it's been given this orphan disease status. And that matters when you're trying to translate therapies. It means that we have a lower bar to pass in terms of preclinical testing, and we can often combine different phases of our clinical trials, like phase 1 and 2, as a single study, which helps to get therapies into clinics faster. What people often don't recognize is that spinal cord injury is the most expensive major medical condition that exists, more than heart disease, more than stroke, more than diabetes. On a per patient basis in direct care costs, it can cost up to $4.5 million to care for someone with a high cervical spinal cord injury. It can cost $2.5 million to care for someone with a mid-thoracic injury, of course, depending on the age at which they're injured. So what that essentially creates in our society is a tremendous willingness to pay. When polled, different insurance companies provided a willingness to pay of over $1 million for spinal cord injury therapy that could be effective for even modest improvements. And so there's this tremendous impetus for us to find something that can help these folks with chronic injuries where they're essentially living with it for life. And when we think about spinal cord injury, I think the first thing that comes to everybody's mind is walking and that silver bullet to get people out of the wheelchair and active and functional again. But actually, when you talk to people with spinal cord injury, what they're looking for are much more subtle improvements. They're looking for improvements in their hand and arm function for folks with cervical injuries, or bowel and bladder and sexual function and autonomic dysregulation. Those are the things they actually put at a higher priority than walking after these types of injuries, because that's what's really affecting their quality of life. So they say, if I could even have a few more motor points of improvement in my grip strength, biceps, deltoid, those types of functions, it could have a tremendous ability for me to be able to hold my children or groom or feed myself. And that's what really matters to folks. And that's what we're really trying to target with some of these therapies. It's important to understand a little bit about the pathobiology of spinal cord injury. And this is something that we've worked on for the last several decades and gotten a better handle on, where everything was originally focused on the initial traumatic injury. And of course, prevention is the most important part. But once that initial traumatic injury occurs, there's a sequence of events that occur afterwards called the secondary injury cascade that can cause as much damage to the spinal cord, if not even worse sometimes, than the initial traumatic insult. So what happens when you initially get that injury is there's this influx of inflammatory cytokines, there's bleeding, there's the beginning of swelling, there's compromise of the small blood vessels supply, the vasculature and the capillaries, and you have cells that are immediately damaged directly by the physical impact, and they begin to release these necrotic cell death products, and they begin to release apoptotic signals that cause other cells nearby to move towards a death pathway. There's also this tremendous infiltration of inflammatory cells that help to amp up this cycle and make things worse and worse. Over time, what that causes is this significant dieback of the axons and the myelinating sheets that are within the spinal cord. So all of the neurons are a wiring system that help transmit those signals. The oligodendrocytes form that myelin or that insulating sheath, and those are very, very sensitive cells that unfortunately die back when exposed to this post-injury milieu. You also have an infiltration of fibroblasts, which begin to deposit scar tissue all throughout the injury site. And you have this constant release of oxygen-free radicals, which themselves can go and damage cell membranes and cause protein coagulation and other issues. And I think we've all heard a lot about astroglial scar in the past, but that's another piece that begins to develop during the chronic phases. So all of these together are working to limit the area in which the injury occurs. That's our body's physiologic response. But in doing so, they actually inhibit the ability of our body to regenerate. And so we and other folks decided that, maybe there's a way we can do something better. Maybe we can actually start to regenerate these injured cords. And a lot of different cell therapies have been tried. So you'll hear about mesenchymal therapies for immunomodulation, or Schwann cells to try and co-opt some of the peripheral nervous system's ability to regenerate and putting that into the spinal cord. But what we and others have really gravitated towards is neural stem cells. And the reason is that neural stem cells are the only cell that can form all the building blocks of an injured central nervous system. They can form the neurons or the wiring. They can form the oligodendrocytes, which are the insulation around those wires. And they can form the astrocytes, which provide the support network for those cells. And no other cell type is able to do that. And that's why I think this is the best way forward if you really want to regenerate the injured central nervous system. So we and others did that. And we originally had good success in animal models, especially in the subacute and early stages of injury. But as we tried to target the more clinically relevant chronic phases of injury, where the vast majority of our patients live, we came up against several barriers to regeneration. And that's really what forms the basis for this talk. So the chronic barriers to regeneration that we found were: one, there's this dense deposition of CSPG, chondroitin sulfate proteoglycan scar, that occurs. So those fibroblasts and astrocytes and microglia that infiltrate, they deposit this dense layer of chondroitin sulfate proteoglycan that itself acts as both a physical and a biochemical barrier to regenerating cells. So when neurons try to grow through it, they just stop. When cells try to migrate across it, they just stop. And that has been a barrier that has plagued our abilities to regenerate the injured CNS for many, many years and affects many other fields. In deep brain stimulation, we get astrogliosis and CSPG formation around the electrodes, which slowly diminishes their ability to deliver current as consistently. We get that after stroke and the marginal rim all around it. And of course, we see it in neoplastic conditions as well, to varying degrees, where you see the gliosis that has occurred around that margin. And the second big barrier that occurs is when you have an injury to the central nervous system, there's a dieback of all these cells. There's now a lack of volume. There's no cells there. And with that, there's no extracellular matrix or support system to help with regeneration and help other cells grow and migrate across. And so you get these small microcystic cavitations that often coalesce into bigger ones. Sometimes they're syrinx sized, sometimes they're just diffuse throughout the parenchyma. But those themselves are another barrier to regeneration. Cells need something to attach to grow, and they need something to attach to stay alive. Without that, they undergo a process called anoikis, where they just begin to die off. If there's nothing to hold on to, they decide that it's not worthwhile and they die off. And the last piece is that the post-injury microenvironment is harsh. It doesn't have all of the normal neurotrophic support that you would find in the central nervous system, typically without injury. And so without that neurotrophic support, the constant growth factors and other neurotrophins that we have to encourage growth and survival of cells, they don't do well. And if we try to transplant things into this chronically injured cord, that lack of neurotrophic support changes their behavior. They're not as regenerative as they might be. And so what we needed is some way to address all of these barriers. And this is what's really been stymieing our ability to regenerate, especially in the chronic injured cord. So we took all of that and we put it together into something called smart cell therapies. And that's what I'm going to talk about today. I'm going to talk about the generation and characterization of this next generation human smart cell therapy that we're hoping can change the way we regenerate the chronically injured CNS. We're going to talk about some of the challenges and opportunities that exist in translation of cell therapies for the central nervous system. And talk a little bit in broad strokes about my vision for the future of CNS regeneration. I do think it's quite bright. I think there is a groundwork that has been laid beautifully before us and some challenges that we can overcome as a collaborative field in order to move some of these therapies to fruition and really help our patients. So let's begin with the generation and characterization of this smart cell therapy. So when we talk about these barriers to regeneration, we said, is there a way that we can address all of them as a single living cell product, a single therapeutic measure to address all of these different barriers to maximize our ability to regenerate? So we have a few of these, and I'm just going to go through them here. So the first one is that neural stem cells, the way we've derived them historically, is they've always had a brain identity. Whenever we transplanted cells into the spinal cord, for example, to try and regenerate the spinal cord, we used cells with a brain identity because that's the way the science evolved. It was all done from original animal models, looking at how do you make a neural stem cell in the brain from the subventricular zone? How does that form different parts of the brain? How does it migrate across the corona radiata? And those are the cells that we continue to make even into the modern day. But the problem is that different cells from different parts of the body and different parts of the CNS have a different signature. And if we're trying to regenerate the spinal cord, we need spinal cord specific neural stem cells. If we're trying to regenerate the cervical cord, we need cervical cord cells. Thoracic cord cells. If we're trying to regenerate the brainstem, we probably need brainstem cells. And the second big barrier that we had to creating a viable therapeutic option for CNS regeneration was poor transplant survival. That lack of trophic support was a big barrier that caused our cell transplants to survive in single-digit percentages usually. 3, 4, 5% of cells would typically survive. The mass majority would die off after the transplant procedure. And we think in large part that's because of this lack of trophic support that exists. And the third big barrier was those extracellular matrix changes that I was talking about, that formation of CSPG scar and those microcystic cavitations. So to address these three, we developed this concept of spinal microenvironment modifying and regenerative therapeutic, or SMART cells. These are a single living cell product, human iPS-derived neural stem cells that we've modified in several key ways so they can address all of these barriers directly. So these smart cells, first, is that they're region specific. We have found ways that are proprietary and now fortunately been published and patented to create stem cells that are specific to what you're trying to regenerate. So if you're trying to regenerate the cervical cord, we can now make a neural stem cell for the cervical cord with that identity. If you want to regenerate the brain, we have a brain neural stem cell. If you want to regenerate the thoracic cord, we have a thoracic neural stem cell. The second key feature of this smart cell package is that they themselves express growth factors necessary for their survival and differentiation to enhance that poor transplant survival that we used to have. And the third big modification is these cells have the ability, either in combination with the way we transplant or just on their own, to modify that extracellular matrix. So these cells express a scar-busting enzyme, which I'll talk about a little bit later, that's able to break down that CSPG scar that's growing all around them and inhibiting their ability to regenerate. And we often transplant these in combination with a self-assembling biomaterial that reconstitutes that lost extracellular matrix so they have something to hold on to and something to grow through. So what we'll do is we'll go through each of these different pieces to show a little snippet of some of the science of how we develop these different things and the rationale for it and what the results were in some of our models. So beginning with the region-specific neural stem cells. I'd be remiss if I didn't say that this is in large part driven by work by Dr. Mohamed Kazai at the University of Toronto. He is a truly exemplary, phenomenal scientist, very, very knowledgeable and a very creative thinker. And this is some of the concept that he came up with for how to regionally pattern neural stem cells so they fit with what they're trying to regenerate. So essentially, this is a basic diagram of some of the regional patterning that occurs as we're developing in the fetal stages. And what happens is there's a series of transcription factors and morphogens that are released that alter the way our brain and our hindbrain and our upper spinal cord and our lower spinal cord develop. And they basically achieve different signatures based on the exposure to those different morphogens: ventralization, dorsalization, anterior or posterior identity. And so we said, well, what if we copy that developmental pathway and use it for our induced pluripotent stem cell derived neural stem cells, the kinds that we can make from any patient or from any other donor? What if we copied embryology over to this new iPS technology? And so that's exactly what we did. So we looked in lots of detail. Sorry, it's just not advancing here. Oops. We looked in lots of detail of what are those morphogens that are actually driving the expression and changes in the identities of these cells, and we copied those essentially into our culture protocols. We took unpatterned neural stem cells, we then posteriorized them, we then cautelized them, and then we more specifically target them to become cervical region neural stem cells, for example. And this is sort of the result of some of that work. So what you're looking at the top is a heat map looking at the different genes are called Hox genes that are basically identifiers of the identity of a particular cell type. So the top few rows here are derived from fetal or adult tissue for the brain or from the derived neural stem cells. So the very top row is brain identity neural stem cells, the typical kind that were made in the lab. And you can see that the heat map there matches the second and third row, which is a fetal or an adult brain-derived neural stem cell tissue. In the fourth row there, the box that's in red, that's our cervical identity neural stem cell. That's the one that we created in a culture dish using these mimicking patterns from embryology. And you can see how closely that matches fetal and adult cervical tissue and how different that is from the brain tissue and how different that is from adult thoracic tissue. Every region has an identity. You can see it in these heat maps. And we were able to recapitulate that identity using these in vitro culture protocols. On the bottom here, you'll see some of the different stains. So we're looking at stains, for example, for neural stem cell identity. That's the nestin on the left and the PAC6. Our cervical neural stem cells retain that identity, but they no longer express OTX2 and FOXG1, which are identity markers for brain-derived neural stem cells. So these are a distinct identity of neural stem cell that's now matched to the area in which we're trying to transplant or regenerate. So far, so good. What we did then is we took those cells and we put them into our models of cervical or brain injury, for example, and we're determining whether or not they keep that identity or being transplanted into that environment changes their identity. So this is a experiment that we did where we injured the cervical cord in animals. We transplanted cortical or brain identity neural stem cells. We transplanted cervical identity neural stem cells, and then we stained them afterwards to look at their differentiation proportion. Oops, I actually don't have the slide, but they retain the same identity that we derived in vitro. So if we put a cervical cell into there, it keeps its cervical identity. If we put a cortical cell into there, it keeps its cortical identity, which is obviously mismatched to the area in which it's going. This is a little bit on our model. So we use a high-fidelity clip contusion model of spinal cord injury. It was developed by Dr. Charles Tatter and Dr. Michael Failings at the University of Toronto. Essentially, during the surgical procedure, we place a modified Segata aneurysm clip around the spinal cord. We snap it shut. That does two things. One, it creates the initial contusive impact that we see in human injuries. And two, it allows you to provide a sustained compression, which is also what we see in human injuries until the point of decompression, for example. And so it creates a very like model to what we see in humans with microcystic cavitation, some of the scar formation. And so these animals, if they're going to recover, that's a big deal because this is such a devastating injury. Any more than this, and they effectively require the equivalent of ICU care. So this is a very severe injury model, much more severe than many impactor models in the cervical spine. And it's a hollow cord injury. It affects the whole cord. And so if you do see recovery, it's a big deal in this model. Just to mention that we also do have a porcine model for a more human-sized spinal cord and physiology. So when we transplant these spinal identity neural stem cells into the spinal cord, what we see is that they actually do better at integrating with the spinal cord tissue of the host and at improving the functional outcomes of these animals. So this graph that you're looking at is a grip strength curve. We're looking at the forelimb grip strength of these animals. The red line is these spinal identity neural stem cells. And you can see how much better these guys do by the end of the experimental paradigm versus the blue line, which is brain-derived neural stem cells. On the right side here, what we're looking at is host and graft cells. So everything with the black dots in it is immunogold staining for human cells. Everything that doesn't have black dots in it is the rodent cells, the host cells. And what you're seeing here is synapse formation between the host cells and the grafted human cells. And it's actually occurring to a much higher degree and with much more robust synapses in the spinal identity neural stem cell group than the brain identity neural stem cell group, just showing some visual evidence of the improved integration of spinal neural stem cells when they're trying to regenerate the spine. So matching the identity of the graft to where it's actually trying to regenerate. So the second important part of this smart cell package is trying to improve this poor transplant survival. The post-injury milieu is harsh in terms of its ability to allow cells to survive, but it's also harsh in what it does to the phenotype of the cells you do transplant and that do survive. So when we put neural stem cells into the injured spinal cord, they often get pushed quite strongly towards an astrocytic phenotype, to the point where 90+% sometimes can become astrocytes. And while we do need a certain number of astrocytes, they're not sort of the pro-regenerative phenotypes that we're often looking for to recover function, things more like neurons and oligodendrocytes. And so what we asked is there some way that we can have these smart cells, these human iPS neural stem cells, express growth factors that they need directly into their local environment to alter their survival and their phenotype. So what we did was we took injured spinal cords and uninjured spinal cords, which intrinsically will express different levels of different factors. We homogenized them, so we get a liquid out, basically. And we exposed our neural stem cells to these to see what actually happens to them, which way are they being driven? And is there a way that we can alter the fate of those neural stem cells by exposing them to different growth factors? So on the left here, We have all the different growth factors that we expose these cells to: BDNF, CNTF, et cetera, all the way down to PDGF. We expose them to either no spinal cord homogenate, injured spinal cord homogenate, or uninjured spinal cord homogenate. And then we saw whether or not their phenotype was altered or reverted by different growth factors. And so what we found is that many of the growth factors had the effect of pushing this fate of the cells back towards the uninjured state, so back towards these pro-regenerative neuron and oligodendrocyte phenotypes. But the best growth factor of all was GDNF. It was best able to push those cells that were exposed to the injured homogenate back to the uninjured state, back to where we want in that pro-regenerative phenotype. And so this is the growth factor that we ended up using. So we took the GDNF and we had it genetically expressed from these smart neural stem cells so that whenever they were transplanted, they would express this GDNF into their local environment and also respond directly to it. And what we found is that the GDNF significantly increased the survival of these transplanted NPCs. Everything that you're looking at in this diagram of the injured spinal cord, everything green is a transplanted human neural stem cell expressing green fluorescent protein. So that way it's tagged and we know it's a human transplanted cell. We got about 3 times the survival out of these GDNF expressing neural stem cells versus conventional neural stem cells, which is a tremendous increase. And what that results in is not just preservation of the graft itself, but preservation of the host tissues. GDNF has an effect on the local tissues as well, the ventral motor neurons that are damaged, the long tracts that are damaged. And what we found is that the GDNF, if you look at these images of the injured cords on the left, is sort of the control with the neural stem cells. You can see how big the cavity is and how devastating the injury is. On the right, you can see what happens when the same stem cells are transplanted, but now they express this GDNF growth factor. You get this significant preservation of the host tissue in addition to survival of the transplanted graft, more preservation of these long tracts after transplant. And so this was very cool to see, is that we're having this double effect where the graft is surviving, but now the host is also surviving better and we're preserving those existing structures. This was not only in some of the long tracts, but we also found this in increased survival of the endogenous motor neuron. So this is chat staining, which looks for acetylcholine. Here, these are images of the ventral motor, the ventral horn rather, and what you're seeing on the bottom is the ventral motor cells. These big, large motor cells are expressing acetylcholine down to the muscle junction. And with GDNF, we're getting a much better survival, that dark brown stain, a much better survival of these very, very important ventral motor neuron cells, the lower motor neurons. There was also an increase in the diversity of the neural subtype populations with, with this GDNF expression. And it's not just immunohistochemical changes that we're seeing. We also saw improvements in the electrical transmission across the lesions with these improved grafts and hosts. So this was a double sucrose gap experiment that we did. Essentially, we put an electrophysiology lead above and below the injury site and look at the conduction through that injury site to see if we can improve that conduction over the epicenter of the injury. And we did. With the GDNF expressing neural stem cells, we're able to see improvements in the amplitude and, to a small extent, the latency across this injury center, suggesting it's not just a immunohistochemical response we're seeing with improved tissue. There's a functional response that we're seeing in conduction of important signals across the injury site. Sorry, I'm fighting a cold at the same time here. So the third part of this smart cell package is this scar-busting enzyme. Chondroitin sulfate proteoglycan as part of the larger glial scar is a real barrier to regeneration, and it's been talked about quite a bit for the last several decades, going back and forth whether or not it's inhibitory or helpful. The general consensus is that initially it's probably helpful to contain and wall off the injury site, but over the long term for regeneration and recovery, it's probably inhibitory because it stops the ability of those cells to migrate in or regenerate past. And so this was all about asking, is there a way we can break down that scar to allow these cells to do what they need to do and regenerate that area? And so many folks came up with an enzyme called chondroitinase, CHABC. That's the middle one in green here. And what they did was they put that into the spinal cords or the brains of injured animals, and they found that it degrades CSPG very effectively, and it's able to break down that scar barrier. And actually, if you put it in combination with stem cells, it does an even better job. The stem cells are able to break past that scar barrier and regenerate. The problem is that it has a lot of off-target effects when you inject it just into the cerebrospinal fluid. So you require fairly high doses to have your effect. And chondroitin sulfate proteoglycan is part of the scar, but it's also part of a lot of the normal structure that makes up our brain parenchyma, our spinal cord parenchyma, the vasculature, the ependymal lining, a lot of different places. And so if you inject large doses of the CHABC directly into the intrathecal space, you get off-target effects, even up to subarachnoid hemorrhages. And so we needed a way to express the CHABC enzyme, this chondroitinase scar-busting enzyme, directly into the local environment where these cells are trying to work, and in some way where it could be controllable. So when we needed the enzyme there to break down that scar, we could do so. And when we didn't need it for long-term, for example, when they were done the regenerative process, we could turn it off. And so we came up with this idea of an inducible promoter. So this is a genetic manipulation of the cells where we take an inducible TET-on promoter, attach it to this chondroitinase enzyme, and in this case also attached it to a red fluorescent reporter. So anytime we give the animals or the cells tetracycline or doxycycline, it turns this switch on and the cells begin to express chondroitinase. And anytime we take away the doxycycline, it turns the switch off and the expression stops. We took that construct, we used a CRISPR-Cas9 technique and inserted it into the safe harbor AAVS1 locus of these human cells. And then we tested them. If we provide doxycycline, the cells turn on. So we see this red expression on the right pane. And if we take it away, the cells turn off and we see this lack of expression on the left pane. So far, so good. For anyone who has an interest in some of the details of all of this, this is what the actual constructs look like, our targeting plasmids, and how we attach them in different ways with different Kozak sequences and other things to enhance their transcription there. Essentially what we did then was we took those cells, we modified them with these CRISPR-Cas9 plasmids to get this insertion into that safe harbor locus. We FACS sorted these cells to find single cells that are actually expressing our construct of interest, and then expanded those expressing cells into monoclonal lines to form our cell bank for the purposes of this experiment. So anytime we generate a new cell line and we've done modifications to it, we have to make sure that it retains the key characteristics of what a neural stem cell should do. So these SMART cells, for example, were able to form neurospheres, primary, secondary, and tertiary neurospheres, which is a hallmark of neural stem cells. They express nestin, a key marker of neural stem cells. And most importantly, they're able to differentiate still into all the key building blocks that neural stem cells should. They can become neurons, which is on the left pane here. They can become astrocytes in the middle pane, and they can become oligodendrocytes. So our genetically modified neural stem cells are still behaving appropriately like human neural stem cells should. We then looked at what is the expression profile from these cells now that we've inserted this genetic switch with the chondroitinase expression. And so the first panel on the left here is qPCR, just looking at the transcript expression by this switch. If we give them doxycycline, you can see the qPCR goes up, it's expressing the transcript. And if we take the doxycycline away, the transcript goes down, meaning we have switch control of this cell. The next three panes, this Morgan-Elson assay, is essentially a effectiveness assay. It's looking at the breakdown products of chondroitin sulfate. So if you take chondroitin sulfate salt and you have it broken down by this enzyme, then you'll get a positive response with these this Morgan-Elson assay. So on the left here, we're showing that it doesn't matter if the cells are differentiated or undifferentiated neural stem cells. We still have doxycycline-based control for effective enzyme degradation of CSPG. In the middle panel, we show that it doesn't matter how many times you cycle, you can turn it on and on again and off again, and it still responds in a very similar fashion, turning on and off its functional activity. And on the very right pane here are the response kinetics, showing how long it actually takes to turn on and then later turn off. So we found that it takes about 2.5 days for it to turn on, reaching full activity by about 3.5 days. And then after that, you can turn it off. It takes about 1.5 days to turn off. So now we know we have functioning cells here. So the next question was, are these cells that are functioning and putting out this enzyme, does it alter the phenotype of the cells and what they're able to do? So this is something called a spot assay. In purple, you have a very dense region of scar called a dense region of CSPG scar, just like you have in the spinal cord. And what you're seeing is these green neural stem cells. They're not able to grow past that purple barrier. They just sort of stop there and they can't go in further. But if we use these SMART cells, these chondroitinase expressing cells, they're able to grow into that scar-like area as if it's not even there. So an indication that the phenotype of these cells has changed and they've acquired a behavior that they didn't have before. So very, very exciting for trying to find ways to break down scar in humans. We also wanted to make sure that they're able to break down scar the way it exists in the spinal cord. So instead of just refined chondroitin sulfate proteoglycan like we use for the spot assay, we wanted to see if it can break down CSPG the way it exists in the injured cord. So we took injured spinal cords. And if it glows bright blue, that means there's breakdown product of CSPG. CSPG is being broken down in that cord and you're staining for that breakdown product. So if we expose that upper cord to chondroitinase enzyme, stock enzyme that you get off the shelf, you can see bright blue all over the place, meaning breakdown is occurring of that ex vivo CSPG. In the very bottom panel, you see typical neural stem cells. It's not bright blue. There's not as much breakdown product. And then you can see our active chondroitinase expressing neural stem cells in the middle panel. It's very similar to that stock enzyme, expressing chondroitinase and breaking down into this breakdown product that we're staining for here. So we know our cells are functional. We know their kinetics. We know they're still neural stem cells. And we know they're able to change the phenotype of these cells and break down actual CSPG that occurs in the spinal cord. The next step, of course, then is to transplant them into a clinically relevant model of spinal cord injury, the one that we talked about before by Dr. Failings. And that's what this paradigm is. It's a lot of information, but essentially, we took adult female RNU, so athymic nude rats, and we gave them either a sham injury on the top row here or a contusion compression injury that we showed. And then we transplanted them with either this active chondroitinase expressing neural stem cell or a control, either a vehicle or a non-expressing neural stem cell. And we expose them to this doxycycline to turn that switch on and off and express the enzyme in vivo inside the animal after the transplant. All of these transplants were done at about the 9 to 10 week mark, so in the chronic injury phase, which is equal to about 9 or 10 months in humans. So by that point, the scar is well established, and these are animals that have plateaued in their recovery, and we don't expect them to spontaneously continue to recover. They sort of are at the function they're going to be at, and that's the end of it unless there's some manipulation done. And that's essentially what occurs in clinic. Those folks who have done the rehabilitation, and we say this is about your new baseline, this is what you can expect for life, that's the level of what these animals were at. And so we transplanted them with these different treatment paradigms, and we also provided several weeks of treadmill rehabilitation, and that's a really important part. A lot of studies that we do in preclinical realm don't involve rehabilitation because it is time consuming and takes a lot of person hours to do. But it's an important thing because obviously our patients receive rehabilitation, and we know that rehabilitation changes the physiology of the spinal cord. It changes the physiology of the patient. If you improve blood flow, you improve cardiorespiratory reserve, you release growth factors with rehabilitation, it's likely to have a synergistic effect with whatever therapy you're trying to deliver, including stem cell therapies. And so we included a period of forced rehabilitation for several weeks for all groups. to see that if that was going to be part of the paradigm in humans, let's make sure we're assessing for those effects in animals. On the right side here is our delivery method. So we essentially did one major injection at the injury epicenter, as well as 4 smaller injections in the rostral and caudal regions on both sides. The rodent spinal cord is organized a little bit differently. The main motor tracks are actually down the middle in the dorsum, so it's a little bit different. They have a dorsal column, but beneath that they have their motor tracks because they are rubrospinal dominant, and so they have a little bit of a different organization and somatotopy than humans. So we put this into our animal models, and we did a lot of different behavioral tests, not just looking at walking, which is an important thing, but looking at things that really matter to patients. So skilled forelimb reaching, the ability to manipulate food pellets in their hand, the ability to reach and coordinate their movements for grooming and all these types of tasks, in addition to sensory changes and the speed and kinematics of which their movements were changing. So a lot of parameters that we find very clinically relevant. And everything in pink is our active treatment group, that active smart cell group. The green is typical neural stem cells without this smart cell set of modifications. And the blue is vehicle control. So basically no treatment. That's their sort of baseline post-injury function. At the very top, you see the sham mean, i.e. an uninjured animal. So what we found is improvements in many different forelimb functions, both unskilled tasks such as grip strength, pure grip strength, as well as skilled reaching tasks, reaching for those pellets or manipulating the food pellet in order to be able to eat it. So very functionally important tasks. And then we sliced the spinal cords to look at it under a microscope in more detail and see what are these grafts doing? How are they changing these forelimb kinetics so significantly? And so what you're looking at here is a sagittal slice of the injured spinal cord. The top is C2, the bottom is about T5. The injury epicenter is marked in the red in the middle there. Everything green is a transplanted neural stem cell. And typically these human transplanted grafts in mouse or rat models, they grow somewhere between 50 and 200 micrometers typically. But we're seeing growth from these cells on the order of 20,000 micrometers, a whole scale difference. They're growing all the way from the cervical medullary junction down to past the mid-thoracic spine, really looking at ways to bridge those long sensory motor tracks that are lost. And that was phenomenal to see, something very, very exciting that we're seeing with these new transplant grafts we're creating. And importantly, that growth that we were seeing was not just haphazard growth. It was actually longitudinally oriented along the dorsal and ventral white matter tracks, growing along those long sensory motor fibers that we're trying to recover and largely sparing the gray matter. So this was, you can't say intentional growth, but this was somewhat meaningful growth from that perspective. The other thing we looked at is something called perineural nets. So perineural nets are a CSPG structure that exists around synapses. Synapses form during early development, and our body reinforces synapses that we use and then prunes the synapses that we don't use. In order to preserve and strengthen the synapses that we do use, it forms these perineural nets. And there's different ways of staining for those, but one of them is WFA, Wisteria floribund, which is everything in purple on the screen. Synapses themselves, we can stain with a marker called PSD95. That's everything in yellow on the screen. And what we found very interestingly is that not only was the CSPG that matters once we turned off the enzyme, not only was the CSPG that matters around those synapses preserved, it was actually increased. We had improved expression of this perineural net mature synapse formation, as well as the density of synapses with this smart cell group in comparison to neural stem cells alone, and certainly a lot more than in the vehicle group. So that was one indication of how this is working. It's growing new long tracks, and it's beginning to form potentially more synapses and more mature synapses with this perineural net formation. And this is just an example. I won't go through all of these in detail, but essentially these human smart cells that we're transplanting, they're developing into this very diverse progeny. You can see mature neuron markers on the middle left. You can see some of the synapses in the middle panel. And these neurons were expressing various types of neurotransmitters like serotonin and acetylcholine, as well as many others, and glutamate. We also wanted to see whether or not these long tracts that were forming from the grafted cells were in fact integrating with the host. So it's one thing to form long tracts, it's one thing to form haphazard synapses, but are these synapses integrating with the host in somewhat meaningful ways? And that's what we were looking at here. So here we did what are called tracer tests. So one of them is on the top right. We injected pseudorabies virus into the muscle forearms, which are responsible for some of the grip, the forearm muscles of the hand. And then we allowed that pseudorabies virus to transmit itself through the neurons as it naturally does. So it starts in the muscle, it heads up the end plate into the neuron. It'll go through the lower motor neuron. It'll then go and jump synapses to the next order of motor neuron in that pathway. And it'll continue to do that until sacrifice to the animal. And there's ways to know how far it's going to go before sacrifice based on existing data of the timeline. And so when we do that tracing, we found that while many of the neurons that are innervating these forearm muscles are from the host, there are in fact connections to those neurons, those lower motor neurons, from our transplanted GFP positive or human grafted cells. So these grafted cells are integrating with important neurons that go down to the muscles. And this may be one mechanism by which they're beginning to cause this regeneration. On the bottom here, you can see a few different examples in our different groups. So the pink arrowheads are pointing to synapses, sort of these fuzzy lines. Those are essentially densities of molecules that hold the synapse structure together and allow it to transmit between different cells. That's between graft and host cells. The black dots are graft cells and the non-black dots are host cells. And what's also cool is that we saw integration of graft and host cells across myelination. So we saw host cells that were myelinated by grafted oligodendrocytes. And we saw grafted neurons that were being myelinated by host oligodendrocytes. So there's real integration, even though they're two different species, there's real integration occurring that seems to be increased when you degrade these inhibitory chondroitin sulfate proteoglycans in the scar. I won't go into too much detail, but we did look in even more detail at the molecular aspects of it because it's a very complex set of regeneration that's occurring. We're seeing lots of surrogate markers that could explain this regeneration, but we want to know at a individual cell type level, how is this regeneration occurring? And so one thing that we did was single cell sequencing to look in more detail at that. So we sequenced host cells and grafted cells from all of our different groups. We then clustered them by species and then clustered them into their individual cell types. And then we could ask specific questions, like if we expose these cells or we have these cells be smart cells where they express this different sets of enzyme, does that alter the ability of these human neurons to grow? And if so, by what molecular pathways? Or does it change the myelination path? And how is it doing so? Is it changing the maturity? Is it changing something in the filopodia or something in the molecular, essentially glues that allow these myelin assemblies to attach? What is it that's actually happening at a molecular level? So we took the cords, we digested them through live cell dissociation, we FAC sorted them into rat cells and human cells. We then encapsulated each of those cells using the Chromium platform, barcoded each cell, took them apart, sequenced them, and then using computational techniques, we're able to tell different species, different cell types, different cell subtypes, and how those are changing based on whether it's a smart cell or non-smart cell. And we have a plethora of data to sort of give us a picture of what's going on here. But just as one example, when we look at human neurons that are grafted, I think the legend might be cut off a bit here, but when we look at grafted human neurons, the red bar on the left there in the violin plot, that's our smart cells, and the one on the right is our conventional neural stem cells. And you can see these smart cells are expressing a lot more of this protein, this GAP43 or GAP43. This is growth-associated protein 43. It's one of the many markers of axon growth. So we see that there's this one potential mechanism by which these human neurons are growing so much longer. It's through this GAP43. On the right side here, we're looking at human oligodendrocytes, and these smart cells were expressing higher levels of aniline, which is an important protein that's responsible for part of the myelin assembly pathway, that insulation forming pathway. So a couple of different insights, and there's quite a bit more data to go through here, which is always exciting to get to tackle. The last part is I wanted to talk about for the smart cell package was that matrix reconstitution. And so this comes back to our barriers, that microcystic cavitation. If cells don't have a place to attach to, there's a lack of extracellular matrix. They're not going to be able to grow through it, and they're not going to be able to migrate or even survive in that area. And so we said, is there a way we can regenerate or replace that lost extracellular matrix, at least for long enough to support these cells, so that eventually the native mechanisms can go and regenerate once those cells have been in place? And so what we migrated towards was this biomaterial called QL6. It was originally made by Medtronic and then was purchased by other companies. Essentially, it's a very simple KK-QL6-KK peptide. So it's a sequence that's arranged in a specific way so that it has specific properties. It has a hydrophobic core and hydrophilic outer sides. So whenever you take this protein, you dissolve it in liquid, it stays in liquid form at room temperature. But then if you go and expose it to the ion concentration of the central nervous system, the CSF, and the natural sodium that is floating in there, you have this principle called electrostatic screening, which causes these basically protein bilayers to assemble into what's called a beta sheet or a fibril. And so essentially the protein, it's liquid, it can be injected, it's inert, it's pH neutral, but once you inject it into the spinal cord, within about 2 hours, it assembles into this beautiful matrix-like hydrogel that we know can support cell adhesion and very much mimics the properties of extracellular matrix. So a very nice replacement that doesn't require us to cut into the spinal cord or put a graft in. It can be injected through a very, very fine needle to minimize the amount of trauma, and it diffuses like a liquid does, but then assembles once it gets into those cavitations. So this is just some of the early work that we did showing that this QL6 biomaterial, when labeled with this green fluorescent marker, it gradually biodegrades over the course of the first 8 weeks, which is an important property that we want. We want the material to go away and be replaced by extracellular matrix. We also tested, and we know that it's pH neutral. Many of our biomaterials that have been used in the past, unfortunately, are quite acidic, which is not great for regeneration. The next thing we wanted to do is the historical use of this biomaterial was with mouse and rat neural stem cells, but moving into the translational realm, we wanted to know whether or not this works with our smart cells, the ones that we're using now. Does this work with human iPS-derived neural stem cells that we anticipate will be the mainstays of regenerative therapy going forward? And so to test this, we took QL6 or plastic bare dishes or Geltrex, which is a mouse tumor-derived extracellular matrix substitute, a standard of cell culture. And we said, can these human neural stem cells plate on this? And what do they do? Is it the same as the standard control, which is Geltrex? Is it worse? Is it better? And what we found is when we follow these from day 1 to day 3 to day 7, you can see that the ones on plastic, some do adhere, but they don't do great. The ones on Geltrex do quite well. You can see quite a broad monolayer adherence in a single plane all along the plate. But the ones on QL6, they actually do something very unique. They attach to the plate, but they form their native conformation. They form neurospheres. They're in 3 dimensions, which they can't do on the Geltrex. And that's actually even better because that's the native conformation of these cells. That's the way they want to be, essentially, in nature and the way we develop. And so we think that that's actually better for their overall proliferation and retaining their phenotype. The Ki67, that middle red stain, that's a proliferation marker. So you can see that on the plastic, there is some proliferation by day 7. On the Geltrex, there's more. And on the QL6, this material, there's even more. So we know that these human neural stem cells do in fact adhere and potentially even make better conformations on this QL6 biomaterial. Just to give a more visual representation of that, this is scanning electron microscopy and transmission electron microscopy to look in more detail. So this is essentially the same thing, but in a much more magnified view, down to even 2 micron scale bar on the very right here. And what you're seeing is the neurosphere in that top left adherent on a big bed of QL6. And as we zoom in, you see the individual cells and these long processes that they're extending out, adhering and attaching to that surrounding QL6. And the transmission electron microscopy in the bottom plane, which is essentially a 2D slice of the same 3D image, just at higher resolution down to the nanometers. You can see these extended processes coming off the cells that are attaching to the surrounding QL6. So we know that this QL6 biomaterial is quite good at supporting these cells. More than just physical support, we want to make sure that it's not altering, that it's actually maintaining the profile of these neural stem cells, because if we're going to transplant it in vivo, we want to make sure it's not changing what these cells do. And so this was an RNA qPCR profiling that we did, looking at 4 different cell lines which were adhered, and then we looked for different markers of neurons and oligodendrocytes and neural stem cells, and essentially we found that the profile when these are plated on Geltrex, the control, is the same as when we plate them on QL6. So it supports the neural stem cell identity, and it doesn't significantly alter it, which is good. So of course the next step then is to transplant these with our human neural stem cells and see, is there a synergistic effect? Are they actually influencing the behavior of the cells or the ability of these animals to recover? And so what we found is that this combination of QL6 with these human neural stem cells, which is that purple line on the top, has a much better improvement in grip strength. And again, these are chronically injured animals. You'll see some of the first assessment points after spinal cord injury are at the 12-week post-spinal cord injury time. So they were transplanted at about 11 weeks, close to 1 year in humans. And you can see that there's this recovery curve that begins very early on and then plateaus around 3 weeks post-transplant. So even in this severe injury model where they've plateaued and there's typically no further recovery, we can begin to induce recovery in the chronic injury state by using the right types of cells with the right types of supports. This is one more functional behavioral assay that we did. So here what we're looking at is a set of kinematics. So we take a high-speed video camera. We put markers on the animals at key joints, so fingers and wrist and elbow and shoulder and trunk. And we look with those high-speed video shots frame by frame. We measure the angle changes and the distance changes to track the movement of that limb and joint through three-dimensional space. And in doing so, we can actually calculate velocities and spreads and different parameters for each of the different joints and figure out how coordinated the movements are. So if the animal is reaching for a pellet, for example, do they reach in a straight line? Is it a very dysfunctional up and down movement where they're doing a lot of corrections? Very similar to a finger-nose test you might do for cerebellar issues. And what we found is with this combination treatment, again in purple, that biomaterial combined with these human neural stem cells, we had improvements in the coordination of these animals. So it's not just that they're stronger, it's that they're functionally improving in meaningful ways and they're more coordinated. And this bore out as well through their locomotion. So this is the composite catwalk index, which is a laser-based measure of their ambulatory ability, looking at the coordination of their hind limbs and forelimbs, their steppage, many other parameters, the speed of their gait. And you can see this purple group did quite a bit better than the other groups, improving upper limb function, improving locomotion. one other question that we wanted to ask was, it's great that we're regenerating these long tracts, but are these tracts actually crossing that lesion epicenter? Or is it more that some are growing down and some are growing up and the ones in the middle are just staying there? Is there actually a bridge that's occurring? And so this is one sort of tracing experiment that we did to answer that question. So we transplanted different colors of human neural stem cells into the injured cervical spinal cord. And so we put green neural stem cells above the injury, we put blue neural stem cells below the injury, and we put red neural stem cells in the middle of the injury. And if these were only growing in one direction, then we should only see green above, we should only see blue below, and we should only see red in the middle. What we found is that there is in fact crossing over. There are cells that are bridging that gap. We saw green cells growing down to the thoracic cord. We saw blue cells that were growing up into the cervical cord, and red cells that were growing in both directions. So some of these transplanted grafts are in fact bridging that gap, which is what we were thinking would happen based on some of the electrophysiology, but it was shown visually here. And so this together is the spinal microenvironment modifying and regenerative therapeutic, or SMART cell package, a way to, in a single living cell product, address all the major barriers that exist to chronic spinal cord injury regeneration and potentially chronic CNS regeneration. And so we're very excited. We saw all of these results and we realized that, we have something here. There's something that is potentially groundbreaking, something that could really help our patients and offer a real therapeutic paradigm. And then the question was, how do we translate that? And I'll just briefly, because we're running a little bit low on time. Essentially what we did was we founded a company. We liberated some of the intellectual property that we had established for some of this work, and we brought it into that company. We sought out investors and advisors, and we were able to begin the early phases of this company and getting some of these cell therapies closer to translation to the clinic. What I'll do is I'm going to end it there because we're at 6 o'clock here, and I'm happy to take any questions.
Please, guys, feel free to ask questions through the chat box. First of all, spectacular seminar, Chris. So this is like a comprehensive approach to treating spinal cord injury, addressing, the stem cells, the barriers, the cystic cavitations, and neurotrophic factors. So usually it's one of these 3 or 4 things that scientists focus on. This is a complete package that would address all these barriers in a comprehensive way and compelling initial results in the animal. So I have a twofold question. First, how, you started off with the company seeking funding for probably translational research and patient-related research. How is that going, number one? Number two, you're looking at an HDE trial, or you're going to look at like a clinical trial, like where you have controls and then the experiment and the intervention group. That's my first part. The other part of the question is, do you envision the need of immunosuppressants with during and after transplantation, and how's that done? Because you didn't use that in the animal, I don't think, but
you're correct.
But yep. And humans, I would suppose, right? Like that's the next, there may need for immunosuppressants, because that's
you're absolutely correct. So in the animal models, we're using a roet-nude rat, which is athymic, so it lacks part of its immune system, and the reason we use that is mostly because this is a xenograft. We're transplanting human cells into a rodent spinal cord, and so there's a greater immune response to that. The question of whether or not an autologous therapy, certainly, or an allograft therapy from another patient requires immunosuppression is actually still an open question. So the majority of trials have used a 6-month period of immunosuppression post-transplant, but the CNS, especially in chronic injury, is considered an immune privilege area. Mm-hmm. And so it's difficult to know whether or not we actually need it. I think most people have done it in trials out of an abundance of caution. At the same time, especially for an autologous self-derived therapy, I think the risk of rejection is significantly lower, and we probably won't require it. But at the moment, that's the sort of standard of care with trials. Regarding how this actually gets processed through the FDA and how we get this into trials with the EMA, et cetera, because of that orphan disease status, we do need to go through a clinical trial process, but we can often combine the trials. So I envision this would be very similar to the previous stem cell spinal cord injury type trials, the early ones, which is a phase 1/2A combined trial, and then essentially safety plus preliminary efficacy, and then a phase 2B3 as a second separate trial. And that's allows us to recruit smaller numbers, have fewer phases to the trial, but at the same time be able to very rigorously study it. It can be done typically against a vehicle control. So one group has the neural stem cells and the other group has a vehicle control, which contain the excipient without the stem cells. The alternate way to approach it is against a historical control. Both seem to be acceptable by the FDA, at least to some extent. It depends on exactly what outcome metrics you're looking for. Regarding the company, we have been very fortunate to have family offices and angel investors that have supported us thus far, as well as some initial grant funding. Right now, we're in that GMP, or good manufacturing clinical grade cell production process. That's an expensive process that we're going through, but that next tranche of funding that's required is for the trial itself, the phase 1/2A, and that's always expensive. Something like this is close to $25 million to run. And so that's where we're looking for right now. Our CEO of the company is a gentleman named Paul Bradshaw. He's a phenomenal CEO. He's a PhD scientist by training, also did his MBA actually at Kellogg. And so he's been leading that charge and raising funds for that purpose, and we're actively raising right now as part of our Series A.
That's supremely impressive, spectacular. Yeah, I'm looking forward to hearing about the clinical trial in your next talk, and truly enjoyable. I had a question. Do you have any questions, Dr. Masnick?
Well, I had a question about what are your thoughts on the applicability of this to, let's say, non-traumatic forms of spinal cord injury, multiple sclerosis or something like that.
Yeah. So this talk is 100%. This talk is focused on what's near to my heart to begin with, my wheelhouse, which is spinal cord injury. But at the same time, we have a vision for applying this to much broader. So we've tried this in cerebral palsy, for example, and we do have good results there. I think it's equally applicable to stroke, traumatic brain injury, many other conditions, especially where there's a focal injury, so that we have a place to transplant it specifically. ALS and MS, I think, are important targets. The difficulty is always that those are more diffuse types of injuries with multiple areas of plaques or degeneration. And so you have to find a way to have these cells get to all of those different spots without causing trauma during the injection. But that's what we're working on right now. ALS, MS, stroke, TBI, and neurodegenerative conditions. All of the above, I think, are important targets for us to go after.
That's spectacular. And then in the chat box, I think 2 questions, similar theme, great work and awesome presentation. In terms of applying the study to humans, will this help acute traumatic injury or other degenerative conditions? Yes. And then acutely, I think, is there a role for acute application, or it needs to be chronic? I mean, this package is for a chronic injury, of course, because of...
Correct. Yeah, it's a much smaller population for acute injury, of course, and there are logistical issues with getting, especially an autologous therapy, into a patient acutely is almost impossible, but a allograft therapy off the shelf can be viable if you're at the right center. The issue is that the milieu after injury is very harsh, and so all of those cytotoxic pieces that you see after an injury that affect the host cells, they also affect what we put inside. And that's been the difficulty in acute injuries. So people have largely shifted towards subacute injuries when the inflammation calms down a little bit. And we do have a separate cell package that's applicable to that time period specifically.
Yeah, I mean, an acute injury, not necessarily, it's not, doesn't seem to be practical, because this patient's going through a lot. They'll be sick, pneumonia, DVTs. I think getting them through this acute injury into rehab and then having this stem cell transplantation, this full package makes much more sense. Beautiful. Well, I can't thank you enough, Dr. Ahuja, for joining us. And we'll hope to host you soon with a clinical topic. Mix it
Thank
up,
you very
clinical and basic science.
Wonderful.
All right. Have a good night, everyone. Thanks for joining us.
Thank you.
Bye.